So today I'll be talking about surface condensation of tau proteins on microtubules. So mainly I'll be showing some interesting results from a set of experiments and motivate the direction in which we're thinking about these. So just to give some background, cells are extremely crowded. There are a few billion proteins and all other kinds of molecules, but it's not a homogeneous sack. They're not well mixed. There is a great degree of spatiotemporal control and compartmentalization. So the question is, how do cells do this? So the traditional way to think about this compartmentalization is that they do this using organelles, which are membrane-bound bodies. So these are enclosed in lipid membranes. So some examples are nucleus, mitochondria. But there's another way to achieve this compartmentalization. So there's recently been interest in this field of phase separation, and so there are these things called biomolecular condensates, which are essentially liquid-like droplets formed out of proteins or RNA. I've just shown some examples here. These ones are in cells, these are created artificially, but the idea is that you have certain molecules, and when the interactions between similar molecules are strong enough to overcome the entropic effects, you get phase separation. So by concentrating certain proteins in a region, you have compartmentalization. So and these are not bound by any membranes, so these are much more dynamic. So in cells, surfaces are quite prevalent. There are membranes, there are polymers like DNA and cytoskeletal network. So these condensates often form on these surfaces and they interact with them. So I've just shown a couple of examples here interacting with the membrane and the chromatin. So the key question we're interested in is what are the physical principles behind this macromolecular condensation on surfaces, especially in context of biological systems. So we'll be focusing on surface condensation of tau proteins on microtubules. This is the experimental system that we're dealing with. So microtubules are really long, stiff polymers. They perform a host of functions, some of which are like providing structural support for the cell, and there are other functions they do. And tau proteins are associated proteins, microtubule associated proteins, and they stabilize the microtubules. So in these experiments, so these were done by a group in IBT Prague. So what they do is, so these are in vitro experiments, they're done out of the cell. So what they do is they take a microtubule, stick it onto a glass cover slip, and introduce fluorescently labeled tau proteins. And now these proteins they condense onto this microtubule, and we can visualize them. So, okay. Yeah, I'll just show this quick video. Yeah, so that's what we're seeing here. These proteins condense on these microtubules and then they stabilize. So this is what it would look like in a more zoomed out fashion. So there are a few So there are a few key features from their experimental observations that we'd like to understand. So let's look at them. So the first one is that if we look at the intensity of these molecules along the microtubules, and we look at the distribution of these intensities, it's a bimodal distribution. Now this is really interesting because microtubules are made up of a single kind of monomer. They're usually thought of as a homogeneous surface. But the fact that there are two distinct states of condensation, it's quite interesting. Another interesting feature is that these, as we had seen in the previous slide, these proteins don't coat the microtubule completely. They form islands, and the fraction of microtubules covered by these proteins is dependent on the concentration. So yeah, it's not like homogeneous binding. So these are quite interesting. And another result is from these experiments where they introduce the tau proteins, and after the islands have formed and stabilized, they wash them out, and this is done repeatedly in cycles. So what we're looking at here is a chymograph. along this direction you have the microtubule position, and this is time. And these light and dark bands that we're seeing are repeated cycles of introducing the tau protein and then washing them out. Now when these tau proteins are washed out, you see the island shrinks, right? And these islands shrink and they'll completely disappear. But what is really curious is that even after the islands completely disappear, in the next round when the tau proteins are introduced again, the islands form at the same location. So there seems to be some sort of memory associated with the system, and these islands are not just forming at random locations. Also, one more interesting thing is that these islands are pinned to the surface, they don't move much. So these are some like interesting observations from their experiments. So we'd like to come up with a simple model to understand what's going on. So now because there's axial symmetry in the system, we can just take a 2D we're working with a lattice gas model, and we can take a 2D model, and like we can call this part as the surface because there's symmetry in the system. And so each site in the lattice is either occupied by the protein or it's unoccupied. If it's occupied, we denote the state by 1. If it's unoccupied, we denote it by 0, and we can write the Hamiltonian. So this accounts for interactions of the proteins in the bulk. This accounts for interactions of the protein when they are bound at the surface. So there's experimental evidence to believe that these tau proteins have enhanced interaction between themselves when they're on the surface. There's something called cooperativity. And there's a term accounting for site-dependent affinity of these proteins onto the surface, and there's a chemical potential which allows exchange of particles with the environment, keeping the concentration fixed. So if we look at, so the site-dependent affinity that we have, if we take a homogeneous affinity, so that's the kind of null model to begin with, because the microtubules are expected to be homogeneous. We do see some similarities to the experimental observations. So I've juxtaposed results from, I forgot to mention, with the Hamiltonian, we're doing Monte Carlo simulations. So these are results from the simulation, and these are experimental results. Again, this is a chymograph. So we're looking at microtubule position and time. And we see these formation of islands and also fusion of islands as seen in the experiments. And there are some other qualitatively similar features to the results, but I haven't shown here. But one key difference is that we don't see distinct states, we don't see islands when we take a homogeneous affinity. There's only a single state of condensation, and this is also reflected in the histogram of it. So heterogeneities in the surface affinities are required. Now these can be, so there are these things called post-translational modifications. Certain molecules can come and bind to monomers of these microtubules and cause heterogeneities in its affinities. So possibly heterogeneities might be created by these. So with heterogeneous affinities, we do get something that's not a homogeneous profile here, but it's still an ongoing work, and I don't have results to show for, I can't make any claims. So, but the key thing is that a homogeneous affinity is not sufficient to explain the data. And heterogeneities are required. So now, I mean, we have a model and we have some observations, and there are qualitative similarities to the results, but we'd like to understand what's the physical mechanism behind this formation of these islands or this condensation. So there are a few theories of surface condensation, but the most pertinent one to these experiments are something called pre-wetting. So the idea is that so this was given by John Kahn in 1976. He gave the thermodynamic theory for this pre-wetting. So the idea is that when you have a solution and molecules and there's some surface introduced, at lower concentrations you have a thin adsorbed layer, and as you increase the concentrations, it will discontinuously transition to a thick condensed layer. So this was, by Kahn, this Yeah. This was by Khan, this was shown to be a first order transition, and this was the phase plot given by Khan. So the fact that there are two distinct states of condensation, and that's what we see in the experiment, indicates that this mechanism of pre-wetting might be at play in this system. Also, like I'd like to point out, I mean, even the lattice gas model that we're using does show in this regime, it shows first order like phase transition. So pre-wetting along with heterogeneities in the affinities might be responsible for these islands that we are seeing in tau proteins. Yeah, so I'd just like to summarize. So there's a physical understanding of this macromolecular condensation is not present. These tau proteins form concentration dependent islands on microtubules. Homogeneous affinities do not give rise to islands, so this is important. So heterogeneities need to be present, even though the microtubule is a homopolymer. And pre-wetting along with heterogeneity may be the physical mechanism behind these formation of these islands of tau proteins on microtubules. So this might be like a nice system to study macromolecular surface condensation in general. So I'd like to thank Zdenek, Marcus, and the students Eva and Teresa at IBT Prague. So they do the experiments, and also Sandeep with who I work here. So yeah, thank you for your attention.
So you have time for maybe one question.
Hi. So if these macromolecular condensates don't form, are there any pathologies which occur in the cell? Is this physiologically relevant?
Yeah. So tau proteins are actually implicated in, so they are actually concentrated in neurons, in axons. And since, I mean, axons are really long and you have the microtubules there. And so these tau proteins stabilize the microtubules. And there are like pathological states where these tau proteins form aggregates. And these have been studied in neurodegenerative diseases like Alzheimer's. So that's why this is relevant, but also it's a nice system to study the physics of surface condensation.
Okay, so let's thank the speaker again.